Hey class, welcome back to another lecture. As I mentioned in our announcements, we are going to combine two lectures in this video. I've kind of cut out quite a bit of information to make this more digestible and a bit more easy for you to understand. So we're going to get into some of the complexities of glucose metabolism, and specifically we're going to start looking at glycolysis, and we're going to look at some of the major enzymes and major intermediates of glycolysis. And we're going to start looking at this in this class because, well, one, metabolism is incredibly important, especially for exercise science. And metabolism and glycolysis is something you're going to come across several times in your future here at HSU. So whether you're taking 379, exercise physiology, or if you're taking zoology, the physiology course that they offer in the zoology department, you're going to be talking about glycolysis. If you take any biochemistry, you're going to be talking about glycolysis. So this is kind of the major hallmark of exercise science when it comes to metabolism and bioenergetics. So what I have for you here on this on this first screen here is glycolysis and all of its beauty. So we are going to discuss. We're going to start at the glucose molecule, and then we're going to go all the way down here to where either pyruvate can enter the mitochondria or where pyruvate will be generated into lactate. So we're going to talk about all these things and get into the nitty-gritty. But before we start with the process, we got to start with understanding carbohydrates. So if we look at this slide here, most of our carbohydrates, well, all of our carbohydrates will come from the diets. So carbohydrates are present in several different forms. You will notice here that we have these polysaccharides. This should be something you are familiar with right now. A polysaccharide is a long chain of repeating links of sugar. So we could see starch is a polysaccharide. So if you're eating potatoes, you're eating brown rice, you're eating quinoa, you're eating buckwheat, these things are going to be complex carbohydrates, which are polysaccharides. And we have these glucose molecules, right? These carbon backbones, and they're basically all bound together by these glycosidic bonds. Cellulose, which is another type of starch that we get from eating plants, another type of carbohydrate, very similar, right? You can see that they're individual micromolecules, right? We're talking about micromolecules again, like we've been talking about since day one. They're all held together by a glycosidic bond, right? Or a bond that is holding glucose together. And then we know that if we're consuming these sugars, I'm just going to keep it very simple and say sugars, they will eventually get converted into glycogen in the liver or converted into glycogen in the muscle. And then you can see that the glycogen molecules are slightly more complicated. So we have these branching chains of these glucose molecules right here, and that makes it a very complex polysaccharide. So when nature creates these carbohydrates, nature does it in a way where these things are held together in these long chains, and they're held together by these bonds. And then when we want to get them into the body, unfortunately, we have to do some work and start breaking these things down, because these long chains of glucose molecules or these carbohydrates can't get past the small intestine. They have to be broken down, and they have to be transported through the small intestines into the blood, and then once they get back into the blood, we will reassemble those glucose molecules into these complex polysaccharides, which we've called glycogen. So we'll talk about that a little bit. There's a couple different varieties of carbohydrates that we have in nature, and we have glucose, mannose, fructose. They're just different types of sugars, and these are basically called monosaccharides because they're single units, right? So if I go back here, these are polysaccharides, right? This is a polysaccharide that is made up of single units of sugar called monosaccharides, right? And monosaccharides are what we like to have pass through the small intestines and get into the body, right? So basically, when we eat carbohydrates, we have to cleave these bonds, right? Chop it, chop it, so that this monosaccharide can get past the lumen or get past the small intestines and into the blood. And we'll talk about that momentarily. So there are several different kind of flavors or versions of monosaccharides. And you don't necessarily need to know these, but just understand that they are the building blocks, right? They are both the building blocks of starch and cellulose and carbohydrates in the diet, right? We can go back to our Lego discussion, right? If I have a single Lego here, and then I connect it to that Lego, and I connect it to that Lego, and I connect it to that Lego, we get something very complicated, right? And these different types of monosaccharides will make up those complex polysaccharides. And again, just notice that these all have a carbon backbone, right? This is why we talked about those carbon backbones very early on in lecture 1 and 2 and 3, right? You can see that they're all having basically a carbon backbone, and these are these are the building blocks that will make those macromolecules. Let me erase this and get that clean, and then we will move on here. Okay, so let's take a look at how these carbohydrates get ingested, get broken down, and then get sent into the hepatic portal vein, where they will be basically determined on how they're going to be used. So when we begin to masticate, right, here's the mouth here, and we bring in, let's say, some carbohydrates. In this case, I have polysaccharides and disaccharides. So what do you think the word disaccharide means? Well, if I have one glucose molecule here, it's going to be bound together with another glucose molecule here, and that's a disaccharide because it's made up of two monosaccharides, right? So if we ingest polysaccharides, like I showed you here, right, these are polysaccharides, basically we're going to start this process of both enzymatic and mechanical digestion. So the first thing that happens is we have these enzymes in our mouth called amylase, right? We know it's an enzyme because it has that ASE. And that amylase is going to be an enzyme that is going to start breaking apart these bonds that belong to very simple sugars, right? So amylase is going to go to work on breaking these bonds up here, right? Now, what do I mean by a simple sugar? Well, amylase doesn't work on everything. So imagine if you went to a restaurant and you ordered a bowl of soup, and then you asked for some crackers with that soup, and they brought you that little basket of those very pale saltines, right? Well, if I put that cracker in my mouth and I did not chew on that cracker would still start to turn into like a crackery paste, right? The reason it would start turning into a paste is because amylase is breaking down those bonds that make that solid, right? So amylase works on some simple carbohydrates. Now, if I let's say I had a sweet potato and I put a piece of sweet potato in my mouth and I didn't chew on the sweet potato, well, amylase wouldn't be able to turn that into a paste. Maybe over the course of 2 or 3 hours it would do it. But the sweet potato, the arrangement of the monosaccharides is just way too complex that amylase really can't. Oops, sorry about that. Amylase really can't do anything to break down that sort of carbohydrate. So we have simple carbohydrates and we have complex carbohydrates, and amylase seems to favor simple carbohydrates. So maybe a disaccharide, it would turn into a paste, but a polysaccharide, it would be unable to do that. So we ingest the sugar or the carbohydrates, and that will go down into the esophagus, down into the lower esophageal sphincter, and into the stomach. Now, if we are eating a mixed meal and we have some protein in the food that we're consuming as well, so let's say we have a little bit of white rice and some protein, well, that protein will release something called pepsin, which is going to basically break down the bonds that are holding amino acids together, right? We also have a little bit of amylase in the stomach and in the pancreas, which is going to continue to break down more of those carbohydrates. And how is it doing that? It's breaking down the carbohydrates by removing these bonds, right? It's just chopping that up. And the reason it's doing that is because once those carbohydrates leave the stomach, right, we have a sphincter here, and it gets into the small intestines. Once it gets into the small intestines, then those monosaccharides can be taken into the blood, right? So we have to break down those polysaccharide complex chains into mono or disaccharides to get them through the small intestines and into the bloodstream. All right. And we call this hydrolyzation. So some of these enzymes like sucrase, maltase, isomaltase, and lactase, right? These are all enzymes. How do we know they're enzymes? Because they all have this ACE, right? These are all enzymes that will break down sugars while they're traveling through the mouth, into the stomach, and into the small intestines, right? So while they're breaking down that sugar, they are hydrolyzing, right? They are breaking down the bonds here and here. Oops, that was too far. And here and here and here. So they're breaking down those bonds into these monosaccharides components, which then can be absorbed into the bloodstream. All right. Now, it's important to know that each type of sugar or carbohydrate, right, I'm just using these interchangeably, is going to have its own special enzyme, which is going to break it down, which means it's highly regulated, and it's going to have its own transport mechanism to get it into the bloodstream, right? So let's kind of talk about this for a little bit. If you look at this description I put here, certain enzymes such as sucrase or maltase or isomaltase or lactase, these are enzymes that are going to break down very specific sugars such as sucrose or maltose or lactose, right? These are all different types of carbohydrates or sugars that we get from our diet. So we have very specific enzymes that will attack those to break those down. And we will also have very specific transporters, which will take them from the small intestines and put them into, I'm just going to draw an arrow here, into the bloodstream, right? So this process of breaking down carbohydrates and getting it into the body so that we could use it as energy, specifically for exercise, is highly, highly, highly regulated. And these enzymes, they're going to hydrolyze these disaccharides, right? Disaccharides, again, means two sugars together, right? Two sugars together. And I'm not going to get into the specifics of these. This is not a nutrition class. We're just talking about how sugar gets in. So these are very generic. Just kind of keep in mind that these versions of carbohydrates, right, are two types of sugars that are bound together, right? So sucrose is a sugar that consists of two monosaccharides, 1 and 2. Maltose is another one that consists of 1 and 2, right? And they're just held together by these bonds. Well, these enzymes are going to come up here and break those bonds so that these are no longer connected and they're just monosaccharides. And the monosaccharides are what's going to pull the sugar, or that's what's going to enter the body, right? So these enzymes are going to hydrolyze those bonds, break those bonds into monosaccharides, and then they can be absorbed. So let's look at that a little bit more. So I want you to imagine for a second that we are, let me just trying to clean this up. We are focusing on this area here, right? The small intestines, the ileum, the duodenum. And I'm going to zoom in, and that's basically what we're looking at here, right? Or here. So if I were to have the small intestines, I'm going to go, whoop, I'm going to draw a circle. Here's a long tube, right? Which we're just going to say is part of the small intestines, right? We know that the middle part, right? This here, the center of that tube is called the lumen, right? The lumen is the center of the tube where things can get into the small intestines, right? And then the small intestines, right? So here it says lumen, right? So lumen is the center of the tube. It's where things are transported into the small intestines. And then we have these this thing called a brush border, right? So inside the lumen of the small intestines, we have these like little finger-like projections that are doing this. And what that does is that increases the surface area of the small intestines so that we can have greater absorption capacity in this particular area, right? So we have them on the bottom, we have them on the top as well. The small intestines kind of look like this, right? These finger-like projections. And then in the center of all that is the lumen. So this picture here and this picture here are essentially the same thing, right? So we'll say that this is the lumen, right? Here's where all the goodies are coming in. And these are called introcytes, right? So these introcytes are a type of cell that is going to allow certain things to pass through them, right? So we'll say here, this brush border, right? Brush border. You can see it here, brush border. This is the introcytes here. So we have all these different cells kind of sitting down here called introcytes. And if you look at this here, here is one introcyte, right? You see that? Here's the second. Let me clean that up. Sorry, I'm drawing with my right hand right now. So here's one. Here's this one here. This one's pretty big. That's another introcyte. And then here's half of one, right? And you can see the brush border here with these little finger-like projections. Well, the reason I'm showing you this is because aligned in this brush border, right, all down in here like this, you see where my highlighter is? And down here, we call these crypts. That's what they're referred to as. We have this machinery, and I'm going to call your attention over here. We have this machinery, which are basically these transporter proteins that will allow certain sugars to pass through the brush border, pass through the introcyte, the cell, right, and then get into the blood, right? So it's a very highly regulated process, and there's lots of things that can go wrong here. Let me clean this up. Okay, so let's look at lactose. Here is a type A of sugar, right? Lactose will be broken down by lactase, and then it can be taken into the introcyte and then into the blood. We have a transporter here called GLUT5, and GLUT5 will accept certain types of sugar. Basically, the sugar will enter GLUT5, pass through the introcyte, and get into the blood. Okay, here we have sucrase, right? That is that enzyme. We have sucrose. Sucrose is going to bind to that enzyme. That enzyme is going to break it apart, and then we'll have two monosaccharides that will enter the bloodstream, right? It'll pass through that introcyte, right, and into the blood. And then we have another one, which is called an SGLT1 transporter. And this thing here will break down or will allow glucose to basically bind to this transporter. And that transporter will carry glucose through the introcyte and into the blood, okay? So when you're looking at this, the big takeaway is, okay, we have the small intestines. The small intestines is basically this circular-like tube. In the center of the tube, we have the glucose coming in, or the lactate, or the maltose, or any type of carbohydrate coming down. Inside of the lumen, we have these finger-like projections, right, which we call the brush border, right? And these finger-like projections right here, you see all those fingers sticking up into the lumen? They're attached to these cells called the introcytes. And these introcytes regulate sugar or carbohydrates coming from the lumen, entering the introcytes, and then entering the bloodstream so that the body can use it, okay? And if we look over here, we can see that the brush border here, right? Let me get a smaller, let's do red. Follow my cursor, the brush border here. When we get down into these crypts right here, right? And right here, when we get down into these crypts, they are basically aligned with all this machinery that will process that glucose, okay, or the lactose, or the sucrose, right, or the starch. We are filled, these crypts down here are filled with this machinery that will basically package, transport, or break down more disaccharides to allow monosaccharides to get into the bloodstream, okay? So that's the takeaway message on that. And if we look at, let's just kind of look at one of these little finger-like projections here, right? So I'm going to say, let's just look at this, okay? We'll look at this finger-like projection, right? This one subunit of the introcyte brush border, because that's what that is. It's an introcyte brush border. So if I zoom in on this one little finger-like projection, this is what it looks like, okay? And this is identified, this one little finger-like projection is called a villus, right? And you can see it here. And essentially, let me just draw here. Let's just say that here is the sucrase enzyme, right? And here is the GLUT5 transporter. And here is the maltase enzyme, which is going to break down maltose, right? And here is, let me grab one more over here, lactase, we'll say lactase, right? Here is the lactase enzyme, right? So those pieces, those machinery, or those mechanisms are all aligned within the CRIP here, right? And when a type of sugar comes in, if it's in a form of a disaccharide, right, 1 and 2, oh, where did it go? There you go. It's going to bind to one of these pieces of machinery, right? If this is an enzyme, this one here, if it's an enzyme, it's going to hydrolyze or break that bond and allow the monosaccharides to enter into the blood, right? Because you can see that these villi, right? This is one villi, it has capillaries in it. And those capillaries are going to take that glucose and allow it to enter the bloodstream, okay? So takeaway message here. Let me go back one more time and just beat a dead horse because I know there's a lot happening here, right? Let me just clean it up. We'll go through one more time. Make sure you got it so that way you don't have to pause and repeat or rewind or whatever thing, anything like that. We have the lumen, which is the center of the small intestines, right? Both below and above the lumen, we have these introcyte cells, right? And these introcyte cells contain something called a brush border, which are filled with villus, right? And that are these finger-like projections here, okay? I'm putting a blue dot on all those little finger-like projections, right? And one single finger-like projection is called a villi, all right? And we know when we have these villi, right? There's sections of them referred to as the crypt, right? So if you think of like horror films like Tales from the Crypt, right? We have these sections here, and in the crypt, we have all of these mechanisms which are going to either transport or further break down sugars that are bound together or hydrolyze, right? Lactase is going to hydrolyze lactose. It's going to break down that disaccharide. SGLT1, which we'll talk about in more detail in a bit, is a transporter. So that's going to package glucose. It's going to send glucose through the introcyte into the capillaries in the villi. GLUT5, this is a transporter as well. So GLUT5 is going to focus primarily on fructose, right? The type of sugar we get from fruit. And that's going to basically allow the disaccharide to bind to GLUT5, and GLUT5 is going to package it and send it through the introcyte into the bloodstream. We have one sucrase. Sucrase is an enzyme which is going to hydrolyze the disaccharide sucrose, and it's going to allow that to be taken into the bloodstream, right? And on and on and on and on. And then we have another glucose transporter here called SGLT1, right? Which is very similar to this. Exact same thing. And that's going to take the glucose and send it into the villi's capillary, which we see right here, and then that is how it's going to get into the hepatic portal vein. So what is the hepatic portal vein? Well, if we look on the next slide here, we can see that the portal vein is right here. So we see the small intestine, which I just showed you, right? And the small intestine, which we can see all in here, is filled with this vasculature system. You see that? I'm highlighting it here, right? There's all these capillaries and vessels here that will allow the glucose to be taken in. Let me make this a little, let's do yellow. It allows the glucose to be taken in, and watch this. Look at where these vessels go, right to this big vessel right here. And look what that's called, the hepatic portal vein. So essentially, all of these capillaries, right? Here's the jejunum, right? This is part of the small intestines. All of these capillaries lead to this big, thick guy right here, which is the hepatic portal vein. And what does that mean? Where is it taking it? Well, if it's hepatic, that means it's taking it right here, which is the liver, right? So let's go to this slide one more time. Let's beat a dead horse. We take in sugar or glucose or carbohydrates from our diet, right? We begin to break it down here in the mouth where we have amylase that is secreted and starts to hydrolyze, right? Hydrolyze some of those bonds that are holding the polysaccharides together. And we know that the polysaccharides, again, are these complex versions of carbohydrates bound together. And we also know that some of these, through the digestion process, what usually makes its way to the small intestines is something like this, right? A disaccharide, two molecules bound together. But when we get to the small intestines, we have machinery there that will hydrolyze that bond, and then a monosaccharide, just one molecule, will make its way into the capillaries. And then that will make its way into the hepatic portal vein, right? So we ingest that glucose, right? It goes down into the stomach. It gets into the stomach. We have some amylase in the stomach, which is chopping up some of that complex carbohydrates. We get down here to the lower stomach sphincter, and then we enter the small intestines, and we know the small intestines has the lumen filled with enterocytes, right? We go back here, we have the lumen. It's filled with these enterocyte cells. These enterocyte cells have a brush border, which are made up of these microvilli, right? V-I-L-L-A. We know that these villi are finger-like projections that reach into the lumen, right? Because the lumen's right here, right? This is the center of that tube. And then we know that within these crypt areas here, of these microvilli, right? We have these mechanisms here, which is going to help us break down disaccharides, bring them into the bloodstream through these capillaries, and then it will enter the hepatic portal vein to eventually get to the liver, and the liver is going to be the base station where it's going to figure out what to do with those sugars and where to send them. One of the things it might do to those sugars is start creating glycogen, right? Especially if you just ate. If you've been eating food and you have insulin being secreted, well, that's going to tell the body that you are in an anabolic state and to start creating glycogen. All right, so now once these monosaccharides arrive to the liver, they're going to need specific transporters to be allowed into the liver, right? So here we can see we have, let's just familiarize you with what's happening here. This is a hepatocyte. That means it's a type of cell in the liver. This is the blood here. Let me change colors. That's not really showing up. You can see that because it's red, right? And we have these different types of sugars here, right? Glucose, fructose, galactose. And you can see that they're monosaccharides, right? So you can see galactose is a type of sugar, right? We get from our diet. Fructose, here's purple, right? Type of sugar we get from our diet. Glucose, a type of sugar we get from our diet. They are going to require specific transporters to get into the liver or the hepatocytes, right? So galactose and fructose, they're going to require GLUT5, right? And glucose is going to require GLUT2, right? So these are really easy transporters to understand, right? And these are transmembrane transporters, right? And we're going to be talking about cell biology pretty soon here. So I'm just going to put T and M for transmembrane. And I'm actually going to throw a picture up for you as well. It should pop up somewhere around here in a second or two. And essentially, if we have a membrane, right? We know that the membrane is made up of lipids, right? And phospholipids. Every cell has a membrane. You guys should know that, right? A transmembrane protein such as this is something that basically will communicate with the outside of the cell. It will go through the membrane, and then it will have a portion inside the cell, right? And that's what a transmembrane protein is. It goes through the entire membrane. So then let's see if I went back to this, then we would have a continuation of the membrane here, right? And here's more of those phospholipids, right? That lipid bilayer, it's right there. So a transmembrane protein then, it's this kind of interruption in the membrane, and it's a different type of material. This is a protein where these are lipids, right? These are phospholipids. So what's going to happen is this transmembrane protein is going to allow a molecule to come in and pass that molecule into the cell, right? Molecules can't interact with these lipid bilayers because they're lipids, right? They're hydrophobic. So things aren't really going to get through there by themselves. So we're going to need a transporter that's going to allow it in. And that's what we see here with the GLUT2, and that's what we see here with the GLUT5. Now, once these sugars make their way into the cell, they're going to interact with a couple type of enzymes, right? And we talked about this a little bit in our last lecture when we were talking about glycogen, but now we're going to do a bit of a deeper dive. So again, we know these are enzymes because they end with ACE, and ACE represents enzymes, right? So we have galactokinase, right? And what that's going to do, it's going to take galactose. Look at here, it has an ATP. It's going to take one of the phosphates from ATP, and it's going to pop it onto that sugar, right? So you see the phosphate here. And then we have this galactose 1 phosphate and ADP, right? And we know that the phosphate was put on the first carbon, okay? Now, if that doesn't make sense, I got a couple more examples for you here, so hopefully it makes sense again, or it will make sense momentarily. So if we look at fructose, okay, we know fructose can leave the blood and enter the hepatocyte through this GLUT5 transmembrane protein. Again, TM, transmembrane, please remember that. Once it gets inside of the cell, here it's, I'm happy to be here. Thanks for letting me in. There is an enzyme waiting for it. It's going to greet it. It's going to open the door. It's going to say, welcome to the liver. We're glad you made it. And this one is called fructose kinase. And again, here's the ACE, so we know it's an enzyme. Well, this enzyme is going to take ATP. It's going to rip off a phosphate from that ATP, and it's going to put it right here on the first carbon of fructose. So then we have something called fructose 1 phosphate and ADP. So there's a bit of an investment phase, right? This is a catabolic reaction because in order for these sugars to get into the hepatocyte or the cells in the liver, we have to have ATP broken down so that the phosphate can be put on the first carbon here or on the first carbon here, okay? We have one more, if we're looking at glucose, right? This is going to be our big focus because we're talking about glycolysis. Glucose is going to enter the transmembrane protein GLUT2. It's going to get inside the hepatocyte. It's going to say, yay, I'm so happy to be here. I made it. there's going to be somebody waiting for it, which is called glucokinase. We know it's an enzyme because it says ACE. Glucose kinase is going to say, hey, glucose, welcome. We're glad you're here. It's going to take a phosphate from ATP, and it's going to put it on the glucose. Now, let me ask you, what carbon did it put that phosphate on? It's not the first. It's not the second. It's not the third. It's not the fourth. It's not the fifth. But it's the sixth, right? Glucose 6-phosphate, and then ADP. So this is how glucose is getting into the liver. This is how galactose and fructose is getting into the liver. Okay, so let me clean this up now. I hope that makes sense. See how we're drawing from previous lectures talking about these carbons? This is why it was so important to talk about carbon numbers. Why is this happening? Well, if these phosphates, we talked about this in skeletal muscle, if these phosphates are not placed on the glucose molecules, the glucose has total freedom to leave. So if a phosphate is not placed here, glucose can say, hey, you know what? I came to the hepatocyte. This is not what I thought. I'm out of here, and I'm going back into the blood. Fructose can do the same thing. If we don't put a phosphate on that first carbon, fructose can say, you know what, this isn't what I expected. I'm not digging this party. I'm out of here. I'm leaving, right? Same thing with galactose, another type of sugar. If we don't put that phosphate on the first carbon, it can say, I'm out of here. I'm leaving. I'm going back into the blood, right? So essentially, when we have these sugars or these carbohydrates, right? I'm just using the words interchangeably. When they enter important cells like skeletal muscle or like hepatocytes, you should be taking notes here, hint, hint, wink, wink, we must trap them. And the way we trap them is by slapping a phosphate on. So now galactokinase, fructose kinase, and glucokinase says, you know what, guys? You're stuck. And then these enzymes laugh with like a sinister laugh saying, now you're trapped. You can't go anywhere. So that makes sure that we keep them in the cell. Now, if this was skeletal muscle, we talked about another enzyme that does something very similar called hexokinase. I'm just going to write H-E with my right hand, X-O kinase that says hexo. Hexokinase is specific to skeletal muscle, and glucokinase is specific to the liver. You need to remember that. Hint, hint, wink, wink. All right, so picking up where we just left off, here you can see a table that's in your textbook. And this basically shows all the transporters that are responsible for allowing glucose or different variations of glucose or sugar to enter specific tissues. So we could see that the GLUT1 transporter, right? And again, let me just refresh your memory here, right? We have these transmembrane proteins. This is my membrane. We know that the membrane is made up of phospholipid bilayers, right? So lots of fat and cholesterol, right? And then we have these proteins embedded inside, and these proteins are going to allow these micromolecules, right? These different sugars, these different carbohydrates to get into specific tissues. And we can see that GLUT1 is found in red blood cells, the brain, the kidneys, the colon, and this GLUT1 is glucose uptake in most cells. So most cells will express this type of transporter that will allow glucose to enter the cell. GLUT transporter 2, this is in intestinal cells. This is in the liver, right? We saw that here, GLUT2, right? It's also in the beta cells of the pancreas where insulin is created. So that has a GLUT2 transporter. Then we have GLUT3, GLUT4, GLUT5, GLUT7, and then here's that SGLUT that we were talking about earlier. And look it's in the intestines, right? I showed you that on this one here, right? Here it is right here. It's in the intestines. So these different transporters, they basically assist in regulating, sorry about that, glucose uptake into the tissue. And there's one here that is, here we go, GLUT5. This is in the small intestines. This allows fructose transport, right? So this one's specifically for fructose. Now, those are transporters that allow glucose to enter most cells. But what I want to focus on right now is GLUT4, right? And GLUT4 is really important because this is what is responsible for letting glucose into skeletal muscle. And we know it's also responsible for adipose tissue as well, which is your fat. We know that this is dependent upon insulin. So insulin is a regulator that has to allow this sugar to enter skeletal muscle. So when we have something like insulin resistance, and we have individuals with diabetes, well, this isn't working and glucose is not getting into the cell. So there's all types of problems that come with that. So let's take a look really quick at insulin-mediated glucose uptake. I want you to remember that. Hint, hint, wink, wink. Insulin-mediated glucose uptake. So I'm going to show you guys the next slide here, and it's going to have a cellular pathway that basically depicts insulin-mediated glucose uptake. So we'll look here. This is a very, very simple rendition of this, okay? But basically what's going to happen is we have these GLUT4 transporters or glucose 4 transporters, and these are the primary mechanism in which glucose enters skeletal muscle via insulin, right? Insulin has to initiate this process, right? And once insulin initiates this process, GLUT4 will be available to take in glucose. So let's say we just ate a meal, and we just here, let's go back to this, right? We just ate some polysaccharides, some carbohydrates. We basically ingested it. We broke it down. We absorbed it in the small intestines. Once it was absorbed, we know that it went into the hepatic portal vein and then to the liver, and then the liver basically decides what to do with it. So let's say the liver takes that glucose and it secretes it throughout the entire body. Well, once that glucose gets to skeletal muscle, it cannot enter that cell or that tissue until GLUT4 is activated. All right, so we have glucose that is circulating in the blood, and we have insulin, which is also circulating in the blood. Why is insulin circulating in the blood at this moment? Well, because we just ate, right? And when we eat, we have insulin being secreted, and we know that insulin is an anabolic hormone, and therefore insulin is going to want to build. And in this case, we are going to want to build in storage. We are going to want to generate glycogen in the muscle, right? So what's going to happen is insulin is going to bind to the insulin receptor, which is right here. And if you look at this receptor, this is an example of I'm going to just write it right here, TM, right? Transmembrane receptor, because it's transmembrane because it passes through the entirety of the membrane, right? So you see how it passes through. We have this section out here, which is going to interact with the insulin. And then we have the section in the cell, which is going to communicate with other proteins in the cell. Now, this receptor is called a tyrosine kinase receptor, okay? Tyrosine kinase receptor. And when insulin binds to this receptor, you see these little yellow balls here? The insulin receptor is going to autophosphorylate, which means phosphates, just like we talk about with ATP, they're going to show up on the receptor, and they are going to activate other proteins. Well, one of the proteins it's going to activate is this IRS1 protein. This is just a relay protein. All it's doing is relaying a message. So here it's turned off, and then once the insulin receptor autophosphorylates, remember that word, it's going to basically put these phosphates onto IRS1, which means that it's turning IRS1 on. IRS1 is now active. It's activated because it has been phosphorylated. So I want you to remember that word, phosphorylated, and I'll type it in somewhere up here. Okay, IRS1 is inactive, but once it's phosphorylated, right, it has phosphates on it, it's turned on. Now, IRS1 is going to turn on something called PI3 kinase, right? So what's going to happen to PI3 kinase? It's going to be phosphorylated. You see, I'm just putting some dots here, right? Meaning that it has a phosphate now. And that protein, this kinase, is going to activate GLUT4. So look at where GLUT4 is in the skeletal muscle, right? When we talked about these glucose transporters in the lumen of, I'm going to clean all this up, in the lumen of the small intestines, where did we find, let me clean all this up, where did we find those transporters, right? So those transporters were transmembrane, right? So you can see some of them are outside of the membrane, some of it is in the membrane, and then some of it is in the cell, right? The intracytes. So what's interesting about GLUT4 is GLUT4 is in the cell, right? And what happens is when PI3K, or just PI3 kinase, the K stands for kinase, PI3K, when this is turned on, this is going to phosphorylate GLUT4. See how I'm putting the P's here, the little phosphates, right? It's going to phosphorylate GLUT4, and GLUT4 is going to do something called translocation. So it's going to move from the center of the cell and then embed itself in the cell membrane. So now it's a transmembrane protein. You see that? So here it's not turned on and it's inactive, but once it's phosphorylated, it's going to embed itself in the membrane, and now it's a transmembrane protein. So once, let me clean this up for you guys. Once it's in the membrane, now glucose can enter the cell, and we can either use it to send through glycolysis, or we can store it, right? And if we use it, we're going to metabolize it. Why are we going to metabolize it? Well, because we want to make some ATP, right? If we don't use it, we're going to store it as glycogen, all right? This is very, very important for you to understand, because this is where the exercise physiology component comes into play, right? We're very focused on how glucose gets into skeletal muscle so that we can metabolize it or we can store it, all right? So we know that it is insulin mediated, right? Insulin has to initiate it. Once insulin initiates this process, the tyrosine kinase receptor, or the insulin receptor, is going to autophosphorylate, right? So these phosphates show up and they bind to the receptor. Once it's phosphorylated, this relay protein, IRS1, is off here, but then it's going to take these phosphates from the receptor and it's going to turn on. And the reason it turns on is because this IRS1 protein has to relay that message to this protein, right? Which is PI3K or PI3 kinase. And then when that message is relayed to PI3K, that's going to phosphorylate. You see the little phosphates I'm putting on there? which means that protein is now turned on and ready to relay that message to GLUT4, right? And then when GLUT4 becomes active, it's going to turn on, and I'm just going to do like this, like, ooh, hey, I'm awake, right? And then it's going to translocate, become a transmembrane protein, and then glucose can now enter the cell to be metabolized through glycolysis or to be stored as glycogen, okay? So this is a cellular pathway for glucose uptake, all right? This is all of the proteins that are involved in glucose uptake. So now that we have an understanding of how sugar gets from our mouths to skeletal muscle cells, we understand that sequence of events. We're going to just talk about the clinical importance of glucose. Okay, why is glucose so important? Well, because for one, it's basically the preferred source of energy for most of the body's tissues, right? Brain cells survive on glucose, right? So if we have an absence in glucose, or if we're not eating glucose, or let's just say we have diabetes. If we have diabetes, well, any number of these things could go wrong. And that's why glucose doesn't enter the cell. Any one of these mechanisms, right, could falter, right, which would cause glucose to essentially not enter the cell and stay in the bloodstream. And then if this cell is not getting in glucose, well, then what happens? Well, then there's little ATP that is being produced glycolytically, right? Which means your metabolism changes. Or let's say you're trying to exercise and there's no ATP in the cell, or you're unable to generate ATP through glycolysis because you have diabetes. And in diabetes, any one of these things could be not behaving correctly. We could have beta cell failure. And if we have beta cell failure, that means that we're not going to have any insulin being produced. And if there's no insulin being produced, well, there's no glucose getting in. There's research out there that shows that insulin resistance can also be a receptor issue, right? We could have a lot of insulin circulating, but not enough of these receptors to propagate the signal. So we could have receptor complications. There could be issues where we don't have autophosphorylation of the receptor. That could be a problem. There have been issues reported with IRS1 and PI3K where the IRS1 is not becoming phosphorylated, or the PI3K is not being phosphorylated. And if those aren't phosphorylated, well, then GLUT4 will never make its way to the membrane. So there's all these complications that can occur with this particular pathway. And this is kind of your first introduction into cellular propagation, right? And that just means that we have a substrate. In this case, we have a hormone. Here we have a substrate that we want to use for energy. This substrate can't get in unless the hormone binds, and we have the activation of these relay proteins, which gives GLUT4 the instructions. It's saying, hey, you can now go to the membrane and let glucose in. All right. So when we have metabolism deranged, like I just said about with diabetes, this is a life-threatening condition, right? So that's what occurs with diabetes, and that's how people become so sick with that disease. Just because one protein, it could be anyone, if this one wants to misbehave, it's going to impact every metabolic process in your body. Just one protein being naughty. And basically, with glucose at rest, normal values are between 70 to 110 milligrams per deciliter. So this is what basal glucose levels look like. After we eat a heavy carbohydrate meal, in a normal person, this could go up to 150 milligrams per deciliter. If you have somebody with diabetes, however, that if they eat let's say they eat a big bowl of pasta, this value, which is normal for healthy individuals, could go up to 400 milligrams per deciliter. And when you have that much glucose in the cell or circulating in the blood, that causes lots of complications, okay? So we reached the point now where we can kind of look at what glycolysis is, because we talked about how the sugar gets there. So then when we get into glycolysis, we're going to basically show you these three different phases of glycolysis. And this is a pathway where we're going to have a sequence of enzymatic reactions that are going to occur that is going to transform the glucose molecule into different types of molecule, which are going to be this 3-carbon pyruvate molecule, right? We know that glucose is a 6-carbon molecule, but once we go through glycolysis, the ultimate goal of glycolysis is to convert this 6-carbon molecule to 2 3-carbon molecules. That is the goal of glycolysis. And in this process, we're going to have an energy investment phase, which we've already kind of talked about a little bit, which means that we're going to lose some ADP. So we have this catabolic step here. And then we're going to have energy generation, where we're going to start to get ATP, right here. And we're going to start to make these electron carriers, which are going to be very important when we get closer to aerobic respiration a couple weeks away, okay? So this is a snapshot of glycolysis, okay? And again, the ultimate goal is to take this 6-carbon molecule and rearrange it through this long series of enzymes to get these 2 3-carbon pyruvate molecules, and I will show you that momentarily. This is going to conclude the first lecture for glycolysis. The second lecture is going to be right behind this. So I'll stop here and let you guys kind of digest this, pun intended, and then we can move into the specifics of glycolysis.